everyone. Welcome to a new session of the paper Molecular Biology. Here we discuss DNA recombination, replica plating and Ames test. The integrity and stability of DNA is essential for the sustenance of life. Recombination is a type of DNA rearrangement which alters the combination of genes present in any individual genome as well as the timing and the level of expression of these genes. Exchange of genetic material in DNA recombination can happen between multiple chromosomes or between different regions of the same chromosome. The recombination takes place between homologous regions of chromosomes. Genetic exchange needs some degree of sequence identity. In diploid eukaryotic organisms, the exchange of genetic information takes place during the process of meiosis. Here, the genetic recombination is to ensure that each gamete includes both maternally and paternally derived genetic information such that the resulting offspring will inherit genes from all four of its grandparents, thereby acquiring a maximum amount of genetic diversity. In a population, this type of genetic variation is crucial to allow organisms to evolve in response to a changing environment. The DNA rearrangements are caused by a set of mechanisms that are collectively called genetic recombination. Recombination lends the genome flexibility. Without genetic recombination, the genes on each individual chromosome would remain fixed in their particular position. Changes could occur by mutation only, which would be hazardous. Recombination provides the means to achieve extensive restructuring, eliminate unfavorable mutation, maintain and spread favorable mutations and endow each individual with a unique set of genetic information. This greatly enhances the evolutionary potential of the genome. Recombination must occur between precisely corresponding sequences in the case of homologous recombination to ensure that not one base pair is lost or added. The newly combined or recombined stretches of DNA must retain their original structure in order to function properly. Two types of recombination can be distinguished. The first one is generalized or homologous recombination and the second one is site specific recombination. In generalized or homologous recombination occurs in eukaryotes during meiosis. A third process, transposition, is a type of recombination in which DNA sequence is inserted into another place without regard to sequence homology. Other applications of recombination include DNA repair, as in the case of double stranded breaks, filling gaps during DNA replication for preventing stalling of the replication fork. Here, normally sister chromatid serves as the donor of missing material through recombination followed by DNA synthesis. Bacterial recombination was experimentally proved by the microbiologist Joshua Lederberg in the bacteria E. coli in 1947. Barbara McClintock and Harriet Creighton in 1931 through experiments with maize demonstrated the role of recombination during the inheritance of chromosomes. Two general models can be distinguished to explain homologous recombination: recombination initiated from a single strand DNA break and recombination initiated from a double stranded break. Types of DNA recombination. Recombination is the production of new DNA molecules from two parental DNA molecules or different segments of the same DNA molecule. Four types of naturally occurring recombination have been identified in living organisms. Here in this figure, you can see the four different types of natural recombination. Let us now study each one of them. First one, general or homologous recombination. General or homologous recombination occurs between DNA molecules of very similar sequence such as homologous chromosomes in diploid organisms. General recombination can occur throughout the genome of diploid organisms using one or a small number of common enzymatic pathways. It can be used to direct error-free repair of double-stranded DNA breaks and generate sequence variation in gametes during meiosis. The second one is non-homologous or illegitimate recombination. Non-homologous or illegitimate recombination occurs in regions where no large-scale sequence similarity is apparent. For example, translocations between different chromosomes or deletions that remove several genes along a chromosome. However, when the DNA sequence at the breakpoints for these events is analyzed, short regions of sequence similarity are found in some cases. For instance, recombination between two similar genes that are several million base pairs apart can lead to deletion of the intervening genes in somatic cells. The third one is site-specific recombination. Site-specific recombination occurs between particular short sequences which are about 12 to 24 base pairs present on otherwise dissimilar parental molecules. Site-specific recombination requires a special enzymatic machinery, basically one enzyme or enzyme system for each particular site. Good examples are the systems for integration of bacteriophage lambda into a bacterial chromosome and the rearrangement of immunoglobulin gene in vertebrate animals. The fourth one is replicative recombination or transposition. Replicative recombination or transposition is a highly specialized form of recombination in which a segment of DNA moves from one location to another, either on the same chromosome or a different chromosome, and generates a new copy of a segment of DNA. Transposable elements or transposons are mobile genetic elements that have selective target sites for insertion and binding. In transposition, transposase, a specific enzyme encoded by the transposon, acts on a target DNA sequence at each end of the transposon. The enzyme acts by disconnecting it from the flanking DNA and then inserting it into a new target DNA site. There is no requirement for homology between the ends of the element and the insertion site. Let us now study about general or homologous recombination. General recombination is an integral part of the complex process of meiosis in sexually reproducing organisms. It results in a crossing over between pairs of genes along a chromosome which are revealed in appropriate matings. The chiasmata that link homologous chromosomes during meiosis are the likely sites of the crossovers that result in recombination. As you can see in this figure, the homologous recombination occurs between similar but not identical copies of the human chromosome 1 during meiosis, which produces new combination of genes. General recombination also occurs in non-sexual organisms when two copies of a chromosome are present. Examples are recombination during F factor mediated conjugal transfer of parts of chromosomes in E. coli, recombination between two phage during a mixed infection of bacteria. As you can see in the following figures, the F factor mediated conjugal transfer of parts of chromosomes in E. coli can be seen. In the following figure, recombination between two phage during a mixed infection of bacteria. The nucleotide sequences are exchanged between two identical molecules of DNA in homologous combination. The most important application of homologous recombination is double strand break repair. During the process of meiosis, homologous recombination produces new combinations and results variation in offspring, which in turn enables populations to adapt during the course of evolution. Exchange of genetic material between different strains of bacteria and viruses through horizontal gene transfer can be used in gene targeting, a technique for introducing genetic changes into target organisms. Let us now study about the different models of homologous recombination. Currently, two main pathways have been proposed how homologous recombination repairs DNA lesions. The double strand break repair or DSBR pathway is used to describe recombination in meiotic cells, predominantly creating crossover products that ensures the accurate segregation of homologs. The synthesis dependent strands annealing or SDSA pathway, on the contrary, typically generates non-crossovers and describes mitotic DNA repair. Homologous recombination repairs DNA in the S and G2 phases of the cell cycle. This is when duplex DNA strands are more easily available and close to each other. Homologous recombination starts with resection of the double strand breakage in the 5' to 3' direction by the MRE11 RAD50 NBS1 complex, forming a 3' single stranded DNA fragment to which RAD52 binds. RAD52 interacts with RAD51, provoking a DNA strand exchange with an undamaged homologous DNA molecule. Assembly of the RAD51 nucleoprotein filament is facilitated by different RAD51 and BRCA2. The single stranded DNA is attached to a ring complex formed by RAD52. This protects against exonucleolytic digestion. RAD52 gene in humans synthesize RAD52 protein, which interacts with RAD51 and replication protein A or RPA and activates RAD51. In eukaryotic cells, RPA binds to single stranded DNA and then binds the 3' overhangs. RPA prevents single stranded DNA from winding back on itself or forming secondary structures during DNA replication. RAD51 and BRCA2 protein forms nucleofilaments, binds single double stranded DNA and interaction with the homologous section on an undamaged DNA molecule called strand invasion. RAD51 will catalyze the strand exchange in a D-loop shape and the DNA synthesis will happen on the invading strand. When this is done, the repaired DNA and the undamaged duplex DNA will change from a D-loop to form a cross-shaped junction, also known as Holliday junction. There are two main models which suggest the pathways the Holliday junction DNA will break into two separate strands: SDSA pathway and DSBR pathway. The figure on the screen shows the SDSA pathway with no crossovers. Complementary base pairing anneals the newly synthesized 3' end of the invading strand to the other 3' end overhang in the damaged chromosome. Remaining flaps of DNA are removed. SDSA pathway finishes with the ligation of single stranded gaps. In the DSBR pathway, the repair process starts by the degradation of the 5' ends of the double stranded break or DNA resection. The resulting single strand DNA ends and then invades the homologous DNA sequences, thereby forming heteroduplex DNA and serve as primers for DNA synthesis. After ligation of the newly synthesized DNA to the resected 5' strands, an intermediate containing two Holliday junctions is formed. Resolution of this joint molecule and mismatch repair results in crossover or no crossover products depending on how the double Holliday junction is cut. The DSBR pathway commonly results in crossover. Chromosomal crossover will occur if one Holliday junction is cut on the crossing strand and the other Holliday junction is cut on the non-crossing strand. This can be seen as shown in the figure along the horizontal purple arrowheads at one Holliday junction and along the vertical orange arrowheads at the other Holliday junction. Alternatively, if the two Holliday junctions are cut on the crossing strands, then chromosomes without crossovers will be produced. Holliday model. Homologous genetic recombination is essential in maintaining genomic stability, such as to protect genomes from double strand breaks or interstrand crosslinks. It is also important in generating genetic diversity that is necessary for evolution. reciprocal exchange of DNA strands between two nearly identical DNA molecules occurs during homologous recombination, results in a four-way DNA junction named as Holliday junction and is named after Robin in 1964. The following figure explains the Holliday model of DNA crossover. In the first step, two homologous DNA molecules line up. Next, the cut in one strand of both the DNAs occur. The cut strands cross and join homologous strands, forming the Holliday structure or Holliday junction. Next, heteroduplex region is formed by branch migration. Resolution of the Holliday structure. DNA strands may be cut along either the vertical line or the horizontal line. The vertical cut will result in crossover between F, F prime and capital F, F prime regions. The heteroduplex region will eventually be corrected by mismatch repair. Finally, the horizontal cut does not lead to crossover after mismatch repair. However, it could cause gene conversion. The Holliday junction is not a static structure. It can undergo spontaneous branch migration in which the branch point can hop forward or backward in a stochastic way. Extensive branch migration requires branch migration enzymes to provide a direction over long stretches of DNA. In between of adjacent branch migration events, the Holliday junction can spontaneously fold or unfold, transforming from one stacked conformer to the other. The Holliday junction can be resolved into two duplex DNA molecules via the action of junction resolving enzymes, and the extent of genetic information exchange depends on the orientation of resolution. T7 endonuclease 1, a junction resolving enzyme, binds to the Holliday junction with high specificity and makes two cuts diagonally across the junction. The Holliday junction plays a large role in genetic recombination. The different arms allow the junction to be mobile. The main purpose of the model is to explain the mechanism of gene conversion during the recombination event. At the location of reciprocal exchange is a distinct marker. Advantage of recombination. Here in this figure you can see the biological roles of recombination. Cell divisions in eukaryotes like plants, animals, fungi and protists that divide through mitosis, where the integrity is maintained by homologous recombination through double strand breaks. The unrepaired double strand breaks can lead to cancer. Tumor suppressor genes BRCA1 and BRCA2 was to play a key role in regulating recombination in humans. Individuals who are heterozygous for BRCA2 are subject to increased risk for breast and ovarian cancer, and loss of both alleles causes Fanconi's anemia. Rad51 binding to single stranded DNA was found to be promoted by BRCA2. This type of recombination brings out diversity in a population. Let us now study about the various applications of homologous recombination. The various applications include gene targeting, protein engineering and cancer therapy. Replica plating. Two types of organisms are present, Auxotrophs and Prototrophs. In the former group, organisms does not synthesize a particular organic compound required for its growth, whereas in the latter groups are characterized by the ability to synthesize all the compounds needed for their growth. Auxotrophs are nutritional mutants, for example, yeast mutant with an inactivated uracil synthesis pathway gene such as orotidine 5-phosphate decarboxylase is a uracil auxotroph. For the growth of such strains, uracil has to be supplemented. In uracil prototroph, such as a wild type strain can grow in the absence of uracil. Beadle and Tatham's Nobel Prize winning work on the one gene one enzyme hypothesis describes the use of auxotrophic genetic markers in molecular genetic studies. If the genetic information of an organism is changed in a stable manner due to mutation, the process is described as mutagenesis. Mutation can result spontaneously in nature. Exposure to mutagens are achieved experimentally using laboratory procedures. The mutagenesis can cause cancer and various other heritable diseases, but it helps in evolution. Hermann Muller, Charlotte Auerbach and J.M. Robson were the pioneers of mutagenesis as a science in the first half of the 20th century. In replica plating technique, colonies were reproduced from master plates choosing any agar-based selective medium without losing the special pattern. The technique was developed by Joshua and Esther Lederberg in 1952 for providing the direct evidence for the existence of pre-existing mutations. This technique isolates both nutritional mutants and antibiotic resistant mutants. This is a widely used method in mutation studies to compare the master plate and any secondary plates and to screen for a desired phenotype. The sensitivity of a particular organism to any substance can be monitored by checking the presence of colony on the master and secondary plates. Replica plating is widely used for screening auxotrophs and antibiotic resistance. Replica plating allows the observation of microbes under a series of growth conditions. The bacteria are grown in an environment that is not selective for a given mutation. This technique is used to transfer the members of each colony to a selective environment. For the convenience, 30 to 3000 numbers of colonies were replica plated due to the difficulty in streaking each out individually onto a separate plate. Method of replica plating. A simple velveteen covered colony transfer device is used to transfer the colonies in nutrient agar medium supplemented with or without a particular antibiotic or nutrient. The fibers of velvet act as fine inoculating needles, picking up the bacterial cells from the surface of this master plate. The velvet with its attached microbes is then touched to the surface of a sterile agar plate, inoculating it. In this manner, microbes can be repeatedly stamped onto media of differing composition. By comparing the presence of colonies following incubation, we can indirectly determine the mutant colonies by their absence in the selective environment. A colony that develops on a complete medium fails to develop on a minimal medium that lacks a specific growth factor. The occurrence of a nutritional mutant is indicated. The microbes that do not grow on the minimal medium represent auxotrophic strains. This method has been applied in numerous experiments to identify the occurrence of mutations. Many of the biochemical pathways in microbes were elucidated in this way by using nutritional mutants. Let us now study about the Ames test. The Ames test was developed in the 1970s by Bruce Ames, professor of biochemistry at University of California, Berkeley, as a fast and sensitive assay of the ability of a chemical compound or mixture to induce mutations in DNA. The Ames test helps to assess the mutagenic potential of chemical compounds. Dr. Ames' work was published as a paper identifying environmental chemicals causing mutations and cancer, which paved the scientists to identify the chemical caused carcinogens in pesticides such as DDT, food additives such as AF2, flame retardant, tris-BP and mutagenic compounds in commercial hair dyes. Salmonella typhimurium was selected to screen defective or mutant genes, making it unable to synthesize the amino acid histidine from the ingredients in its culture medium. The assay is based upon the reversion of mutations in the histidine operon in the bacterium Salmonella typhimurium. The histidine operon encodes enzymes required for the biosynthesis of the amino acid histidine. Strains with mutations in the histidine operon are histidine auxotrophs. Revertants that restore the histidine plus phenotype will grow on minimal medium plates without histidine. This provides a simple sensitive selection for revertants of his mutants. The histidine mutants are mixed with the potential mutagen, then plated on a minimal medium with a very small amount of histidine. The concentration of histidine used is limiting. So, after the cells go through several cell divisions, the histidine is used up and the auxotrophs stop growing. However, if the potential mutagen induces his plus revertants during the initial few cell divisions, then each of the resulting revertants will continue to divide and form a colony. The number of colonies produced is proportional to how efficiently the mutagen reverts the original mutation. Ames test bacterial reverse mutation assay. The bacteria reversed mutation assay is used to evaluate the mutagenic properties of test articles. The test uses amino acid dependent strains of Salmonella typhimurium and Escherichia coli. In the absence of an external histidine source, the cells cannot grow to form colonies. Colony growth is resumed if a reversion of the mutation occurs, allowing the production of histidine to be resumed. Spontaneous reversions with each of the strains mutagenic compounds cause an increase in the number of revertant colonies relative to the background level. Let us now summarize what we have learnt in this session. Genetic recombination and recombinational DNA repair are two facets of a universal process that is essential for genomic integrity. This process, termed homologous recombination, occurs when a broken or damaged chromosome uses a second homologous chromosome as a template for its repair. In meiotic cells, double-stranded DNA breaks are programmed and the ensuing recombination serves to link homologous chromosomes via crossovers to ensure their proper segregation during the first meiotic division. In mitotic cells, recombination is used to repair accidental DNA damage, typically engaging the sister chromatid as the repair template and minimizing crossover formation in order to maintain chromosome stability. Recombination is an elaborately orchestrated process involving dozens of proteins and strict regulation to avoid interference with other DNA metabolic processes, the formation of potentially toxic intermediates and the generation of chromosomal rearrangements. Recombination DNA repair is a universal aspect of DNA metabolism and is essential for genomic integrity. Replica plating is a technique used mainly for identifying and selecting mutant colonies of a microorganism when the mutant can only be selected against. For instance, nutritionally defective mutants can be detected by replica plating. Ames test serves as a quick and convenient assay to estimate the carcinogenic potential of a compound because standard carcinogen assays on mice and rats are time consuming and expensive. Here are a few questions for you to work out. 1. Discuss enzymes involved in homologous recombination. 2. Elaborate the technique of gene targeting. 3. Describe DNA repair mechanism. 4. Discuss the relevance of replica plating. 5. Discuss the clinical significance of Ames test. 6. Elaborate molecular mechanisms of recombinational DNA repair. Here I can suggest a few books for your reference. Genes 8, written by Benjamin Lewin, 2004, published by Pearson Prentice Hall, Pearson Education Incorporated. Laboratory Exercises in Microbiology, 6th edition, written by Prescott M. Lansing, Harley P. John, Clean A. Donald, in the year 2005, published by McGraw-Hill College Division. Biology of Microorganisms, written by Madigan and Martinko Brock, published by Pearson Prentice Hall. Lewin's Essential of Genes, 3rd edition, written by Jocelyn E. Krebs, Elliot S. Goldstein, Stephen T. Kilpatrick, page number 850 and 940. an improved bacterial test system for the detection and classification of mutagens and carcinogens, published in the journal PNAS by Ames B., F. Lee and W. Durston in the year 1973, page numbers 782 to 786. Thank you for watching this program. Let's meet again. Until then, bye.